Okay. So, uh, so now as I told you, whatever the DNA is there, okay, that is recombinant DNA, that needs to be separated, right? So for the separation, what is the technique which we use is the electrophoresis. Okay, so the basic electrophoresis that we use over here is the gel electrophoresis. Electrophoresis, right? So usually the DNA, it has what? Negative. Oh, just a second. This. Yeah. So usually the DNA has what? Negative charge on it. Okay, so here during the separation, during the gel electrophoresis process, negative charge helps in the separation of the DNA fragments, because this cassette is having what? A cathode as well as anode. So probably what will happen is the DNA fragments, they will move towards the anode whenever an electricity is being applied. Okay, so as I already told you that whatever the gel which we use is the polyacrylamide gel, and for the identification purpose you use the ethidium bromide, right? And then you see it under the whatever the computer is there, on that you will get the results. So usually whatever DNA strands you get over here, for example, if I want to take this molecular size DNA, what I'll do is I'll cut this piece, okay, and then that process where I isolate this particular band of the DNA, that's called as what? Elution. Okay, so the process in which your preferred DNA bands have been sliced off from the agarose gel, they are called as what? Elution. Okay, so this is a very useful technique for defining whether it's a recombinant or a non-recombinant DNA. Okay, so now as I already told you that if you consider a tool of a cloning vector, now cloning vector means identifying identical copies, right? So for the identical copies, you need three things. One is the origin of replication, then you have a selectable marker, and then the presence of the restriction site. So let's see one by one. Now, origin of replication, I have already told you what it is, right? So now let's go to the second thing, that is the selectable marker. Okay, now for each cloning vector, okay, there must be a selectable marker. Then only you can differentiate between which is the recombinant DNA and non-recombinant DNA. Now there is one more term over here, okay, which is called as transformants and non-transformant. Means recombinant DNA is for the transformant and non-transformant means non-recombinant DNA. So if you use a selectable marker, then what will happen is you can identify and eliminate that particular non-transformant as well as a transformant DNA which is there, right? So the process of doing so is called as what? Transformation. Transformation. Okay, so this is a process by which you take a piece of DNA and introduce into your host bacterium. So usually there will be a gene coding sequence. Okay, for example, say, let's take some antibiotics. Okay, so for the antibiotic sake, you are doing this particular procedure. Okay, and what happens is there you will find, or you have to identify what? A gene coding resistance. So whatever the gene which codes the resistance, for example, we have different kinds of antibiotic like ampicillin or penicillin, tetracycline, right? So these are what considered as a selectable markers over here. Now these are present. Why? Because it will not allow any other undesirable gene to come ahead. So only whatever you need, that will be presentable. So for that reason only selectable marker gene is very important. Okay, so usually whatever the normal E. coli cells which is there, they do not have any resistance gene of the antibiotic. So you should insert it and then you have to run the particular procedure. So that is the role of what? The selectable marker. Then next you have is what? The presence of restriction site. Now, there are certain characteristics that you should follow in order for your restriction enzyme to cut at that particular sequence. Okay, which I already told you that if you're taking, suppose I gave you an example of echo R1, right? So echo R1, if you're taking it, just cuts on that particular palindromic sequence, that is G and A. Okay, between G and A, it will cut. Okay, so now what are the characteristics you need to know for the restriction enzymes to act on it? So the first thing is the size. It shouldn't be more means it shouldn't be too big in size, neither too small. Okay, then the integration of the donor DNA should not interfere with what the replication procedure and the property of cloning vector. Means you have two things over here. One is what your host DNA. Then you have a template DNA. Template DNA means what, the copy which you have already, which you're going to use for the further procedure. So whatever DNA you're taking, the donor DNA, it should not interfere rest of your procedures. Okay, so that is one important point. Third one is what, there should be several sites for cloning. It's not that only one site will be there for the cloning. So it will become difficult for that process to complete. So there should be several sites for cloning. Next is what, both vector and sample DNA. As I told you, they are digested with the same restriction enzyme. This yesterday also I told you that the restriction enzyme should be same for the vector as well as for the sample DNA which you're going to isolate, and then you have to combine it and grow into a bacterial host. So that particular vector is going to contain whatever your suitable selectable markers are there, so that it will run correctly. And lastly, a vector need not necessarily have components that are helpful in target gene expression. Okay, means what? Target gene expression is what? Obviously, it should have a particular target. Okay, if it will go to one, or if say suppose if there are two similar sites, okay, and only just a little bit difference is there, and you're putting that particular vector, and vector is not knowing that which site has to get targeted. So then what will happen is, again, it will make that process complicated and the procedure will not occur up to date. So for that, what you have to do is, you should take care that whatever vector you're going to use over here, it should be target oriented. Okay, and accordingly, it should express itself. So only if these things are present, then only the restriction enzymes will act on what? The restriction sites. Or you can also say it as what? The cloning sites. Okay, then next one is the types of cloning vectors. Now, when you come to the cloning vectors, okay, now usually we already know that according to the origin of replication and the selectable market, you choose what type of vector is being required over there, right? So usually cloning vectors are what they are. Now, for example, I'll just, okay, I will start from here only. Okay, so plasmid. Okay, that's the first one. First type of cloning vector is what? The plasmid. So now what is a plasmid? A bacterial cloning vectors. They are called as what? Plasmid. That is the double cellular circular extra chromosomal material is called as the plasmid. Okay, so they will be having a particular site where you can insert this DNA fragments, and then you will use the restriction enzyme which will go and play its role on particular restriction sites. Okay, so a typical plasmid, if you take, it's 4361 base pair in size. Okay, and whatever the cloning range which you can insert into it is 0.1 to 10 kilobase. Okay, so usually means most of the time, whatever the plasmid is there, that particular gene is isolated from what? E. coli. That's why I gave you the common example is what? ECO R1. Okay, so that may have what? Any of the antibiotic resistant gene which you can use. Another one is the bacterial artificial chromosome. So in the name itself, it's there. A bacterial artificial chromosome means what? Which you have isolated from the bacterial cells. So every cloning vector has what? Different size and different capacity to intake your gene. So accordingly, you have to select it. Third one is the yeast artificial chromosome, which is obviously derived from the yeast cells. So this YAC is mostly used in the mapping and sequencing of the genome. For example, the HGP, which is there, that is a Human Genome Project. Okay, there you can use this BAC, YAC, all these things. Okay, so usually the if you see the size over here, now this was 4,361. Okay, this is what 11,827, whereas your YAC it's 11,400. So if you compare mostly all the things, okay, out of this three, this thing the BAC has what, it's bigger in size, so you can incorporate a little more also in that particular means gene inside that particular plasmid. Okay, so whatever plasmids you're going to take, according to that only you have to choose what, a marker. So whenever you're writing the points, okay, regarding the marker, it's essential to write selectable marker for the plasmid. Like for example, here how what is written, the marker is comparable to the yeast. Okay, so in the same way, if you take bacteria, that will also be comparable to the bacteria itself. So from which species you're going to isolate that particular thing, that particular selectable marker you will be using. In the same way, bacteriophage. Now, bacteriophage is particular virus, okay, which usually what it does is it enters into the host. Okay, it will degrade the host DNA and develop itself. So that's called as a bacteriophage. And then you have a combination of the vector, which is called as what, plasmid vector. So it's basically a combination of your bacteriophage and the plasmid. So these two together are called as what, plasmid vector. Next you have is what, human artificial chromosome. Now for the human genes, okay, for the transcription, whatever it's required for the transformation process, there you use what, human artificial chromosome. Okay, so whenever you're using the vectors for the clone genes, if it's, these are all particularly for the humans, you can say. In the same way you have for the plants also. Okay, so if you take for the plants, basically you have agrobacterium. Okay, so there is a microorganism called as what, agrobacterium. So this agrobacterium is nothing but a pathogen. Okay, so usually in the dicot plants you will find this agrobacterium. So this has the capacity to deliver your tDNA, that is your transfer DNA. Okay, so what happens is this dicot plants, they develop tumor. So in order to avoid the tumor, what you can do is directly you can produce this tDNA and then you can transfer it, and then next plants, whatever will grow, it will be free from that particular tumor. Okay, so that is the use of the plasmid. You can just, what do you say, rectify the whatever mistakes are there or limitations are there for a plant growth, and then you can produce a new plant accordingly. So whatever tumor inducing thing is there, it's called as what, TI. Okay, and this transforms what, your tDNA. In the same way for the animals, retroviruses which are there. So retroviruses has the capacity to convert what, your normal cells to the cancerous cells. Okay, so hence the retroviruses, if you are using it to deliver the desirable gene into an animal cell, you have to make sure the cancer causing capacity is being what, disarmed over there. So if you're not locking that particular cancer producing what do you say, facility of this retrovirus, and you use it for a gene transfer into an animal cell, then obviously that animal is going to get what, cancer. Okay, so these are what, certain cloning vectors and its types. Next is the process of the gene cloning. Just a second. So before going to the process of gene cloning, okay, now there is one more thing which is called as competent host. This is also an essential tool for the recombinant DNA. Okay, so usually whatever your DNA is there, it is not able to pass through the, what is a cell membrane. Okay, so hence what happens is you have to make your bacterial cell, make bacterial cell competent. So this is only called as what, competent host. You have to make your bacterial cells to take up that particular DNA which you want to insert. So for doing this, there are several methods which you can follow. Okay, so in that, the first method which you can use over here is what? You can treat them. For example, treat it with a particular cations. Now, particular cations means what? Calcium is being used over here. So if you treat your bacterial cell with the calcium, it will automatically increase the efficiency to uptake the DNA into that bacterial cell. And then what you can do is whatever your rDNA is there, that can be forced into this bacterial cells. But for that, you have to do what? Incubation. So incubation is nothing but whatever your bacterial cells are there, you have to keep it under sterile conditions for a required amount of temperature, pressure, humidity, whatever is there. And then what will happen is it will be forced to take that particular recombinant DNA into it. And once it takes that particular recombinant DNA inside, you have to make sure whatever the cell membrane is there, it closes. So to close it, what you have to do, you have to put it on the ice. So when you put it on the ice, whatever the pores of that cell membrane is there, that will close. And then what will happen is the recombinant DNA is safe inside your bacteria. Okay, next what you will do, whatever recombinant DNA now it's there inside your bacterial cell, you just have to inject it. Inject it into whatever nucleus is there of animal cell. Now this process is called as what? Micro injection. So there will be a gun, okay, which is basically called as a gene gun. Okay, that whatever your gene is, that will be present. So with a very high velocity, you just pull that particular gun, and then what will happen is it will just go into that particular nucleus of the animal cell where you want to insert it. So most important thing here again you have to take care is what? If your whatever bacterial cell is there, it's pathogenic, means if it's causing any disease after insertion into the animal cell, make sure to disarm that particular property. Okay, so whatever disarm pathogens are there, that you can use as a vector to infect the cell and transfer the recombinant DNA into your host. Okay, so this is what is called as a competent host and how you actually insert a recombinant DNA. Okay, so now the next part is the process of recombinant DNA technology. That is, in detail, I'm going to tell you that how actually the rDNA technology works. Okay, so for this, you have certain points, okay, which is very important. Now, as I told you, PCR, okay, the polymerase chain reaction, that is also an important point of the recombinant DNA. So one by one, I'll just tell you. Now, already you know the basic points of isolation, cutting it, and all those things, right? So I'll just write it down. If they ask you about the process of rDNA technology, then these are the points you should write. Okay, so first thing that you have to write is what? This is going to take place at a specific sequence. Okay, so that is the first point that you have to write. Afterwards, the steps like isolation of the DNA, means whatever your desired DNA is there. Now see, it's not simple to isolate the DNA. Before isolating the DNA, or in order to isolate it, what you have to do is you have to break the cells. Okay, after you break the cells, the DNA will get released. Okay, and then along with the DNA, because in a cell many other things are also present like RNA, proteins, right, lipids, all these things are present. But your main part is what? Isolation of the DNA. So for isolating the DNA, what you will do, first of all, you will break the cells. Then what you will do, you will remove all the unwanted thing, materials, right? So because see, now basically to do this, what you can do is treat the cells again with certain enzymes. For example, there are plenty of enzymes like lysozyme, or you can take, if it's a plant cell, cellulase is there. If it's a fungus you're acting on, then you have chitinase. Okay, so these are the enzymes which is going to act on the cell wall. See, because the plant cell, it contains what? Cellulose. So cellulase enzyme you're using. Fungus contains chitin. So you use this chitinase enzyme to break the wall, whatever is there. So then what you do is, after removing, or means after breaking the cell wall by using any of this, you have to remove the unwanted materials. So if you want to remove the RNA, you have something called as ribonuclease. Ribonuclease. That's the enzyme where you can remove the RNA, and if it's for the proteins, it's protease. Right? So finally, whatever your purified DNA is there, that you have to precipitate it out, and you have to make sure that you have to put it into what? Chilled ethanol. Okay, so whatever purified DNA is there, you have to add the chilled what do you say, ethanol, that is nothing but the alcohol, and then you will see small fine threads of the DNA in your chilled ethanol. Once this is done, you go to the second procedure, which is cutting of the DNA. So I'll just write it over here. Second point, that is what? Cutting of DNA. Now, cutting of DNA where? At specific location. Okay, so in the first step, what you have done is you have selected a particular cell. Okay, you are breaking it down by using certain enzymes like lysozyme, cellulase, or chitinase, and you're removing the unwanted materials such as RNA, proteins, whatever is there. Now, whatever purified DNA is there, that you are going into that, you're going to add the chilled ethanol, and then you can see the fine threads in your suspension. Secondly, after this, what you do is you cut the DNA at specific location. Now, here comes the role of what? Restriction enzyme, right? So, in restriction enzyme, what it will do is it will perform like various digestion of that particular DNA. So, digesting the DNA with what? Incubation. This is very important, okay, because you're doing everything artificially, so you should give the proper conditions for it to take in the DNA, replicate, and all those things. So you will incubate that particular purified DNA, and you will treat it with the restriction enzyme during that time. So you're going to maintain what? Certain optimal conditions which are there. After maintaining certain optimal conditions, what will the restriction enzyme do? It will go and cut the specific site of that DNA fragments, and you're going to get DNA fragments which is cut at the specific location. Now you know what happens next. You have to isolate it. So again, you will undergo through all the processes which I have told you earlier, that is the gel electrophoresis. Now, as I told you, that DNA is negatively charged. So hence, what will happen is separation will be towards the anode because it's a negative charge, and depending upon the molecular size, that is the larger fragments will be first in that particular gel electrophoresis over here, and then you will get the smaller size fragments. So accordingly, when you expose it to the UV radiation, you can just isolate it in the form of the elution, right? A piece of that particular gel you can cut and you can isolate it. That you are doing it through what? The gel electrophoresis. Now, once that particular process is done, now you are just going to get a one small, what do you say, copy maybe of that particular band. But that is not enough for your entire procedures, because you have to maybe run that particular procedure plenty of times. So every time you cannot isolate it and then again use it and do the same repeated thing, because it is a loss of money as well as loss of time. For that only you have the third step, which is called as what? Amplification of gene. So how do you amplify gene? PCR, that is the polymerase chain reaction. So whatever gene is of your interest, just make multiple copies like the Xerox sets, whichever is there, and then you're for doing this, what you use over here is, one is called as a primer, and second is a enzyme which is called as DNA polymerase. Polymerase means what? Chain, right? So by using the primer as well as the DNA polymerase enzyme, you are going to carry out this polymerase chain reaction to get multiple copies of your interested gene. Now, what is this primer? Okay, so primers are nothing but they are actually very small, but they are chemically synthesized oligonucleotides. We know, right? Mononucleotides, dinucleotides, polynucleotides, like that. So these primers are what? They are nothing but chemically synthesized oligonucleotides. So these primers will be complementary to your to the regions which are already present in the DNA. So because of that only you use this what primers over there. Okay, now whenever you're running a polymerase chain reaction, you have three major steps in this. Okay, so the first step which is there is the denaturation. Okay, then second one is the annealing, and third one is the extension. It's very simple. Denaturation means what? You have to heat your target DNA so that you can separate the two strands of DNA, right? So you already know that this is the DNA. Now you have to separate these two strands, right? So for that purpose, you denature your DNA at 94 degrees Celsius, and then your two strands will be separated. Secondly, what you will do, now we have separated strands. Now I told you we are adding the complementary thing, which is a primer. Now annealing means nothing but sealing. Okay. So here what you do is you basically pair that particular primer, okay, with your complementary base sequence. So this two strands, out of that one strand you will what do you say, fuse it with the primer, and then whatever after fusing you will extend that particular complementary strands of the DNA from the end 3 prime OH. So for extension, what you use? DNA polymerase enzyme. And then you get what? A new DNA strand. New DNA strand. So that's it. You have completed the process of replication of the DNA. As much as number you want, you will get the copies of it. And then what you can do is you can isolate it later and use it for your purpose, whatever purpose you're using it for. That is nothing but the cloning, right? So after cloning, wherever you have to insert it, you can do it. Next, after this, the next procedure is what? The fourth step, that is just insert it into the host. So how to insert it? Already I told you, competent, make it competent, insert it, okay, in whatever cells, if you're taking bacteriophage, a plasmid, plasmid, whatever it is. After inserting, the fifth step is what? You have to obtain your foreign gene. Obtain the foreign gene product. So once you obtain the foreign gene product, okay, see now here everything, the conditions should be properly maintained. Then only desirable proteins will be synthesized and you will get a desirable what? Recombinant protein. Then what you will do is you will go to the last process, which is called as the downstream processing. Okay, so this is a very important part, which is nothing but the downstream processing. So downstream processing means usually what? It's the separation of your purified product. Okay, so whatever purified DNA you have taken, right? So that you're going to what? Let it go under a downstream processing. Now, in order to do the downstream processing, you will need different types of, like, there are certain bioreactors like this. As you can see over here, bioreactors will be present because the product has to be what put or formulated with suitable preservatives. Then only it can be preserved. Okay, for example, if it's a drug or something else, obviously you have to preserve it to increase the shelf life. So for this downstream processing and all those things, what you use is a bioreactor. So as you can see over here, this is the two kinds of bioreactors which you can use. So bioreactor is simply nothing but a cylindrical vessel where you put all your biological processes, but this is used industry on an industrial scale. Okay, so here only you multiply whatever your recombinant cells are there in a particular culture system. Okay, then you get a fresh medium, then you get fresh cells. Okay, you add different kinds of what do you say, raw materials into it. For example, it may be the plant cell, animal cell, or human cell, wherever you want to insert this particular desired DNA. So bioreactor usually is very good, what do you say, machines. They are very good machines because they provide that optimal condition where you can achieve your what desired product. So what are the optimal temperature which it takes care of is the temperature, pH, the substrate which you're going to add, then different kinds of salts or whatever gases like oxygen, vitamins, and all those stuff. So for doing this, there are two kinds. One is the stirred tank bioreactor and a sparge stirred tank bioreactor. So stirred tank bioreactor, usually it's cylindrical, as you can see over here, curved base it is having. So here what will happen is it will facilitate the mixing of the whatever contents are there by using a stirrer. So hence the name is called as stirred tank bioreactor. But in a sparge stirred tank bioreactor, along with that particular stirrer, you will be providing it with what? Sterile air inside. Okay, so basically those bioreactors are what? Agitator systems, where it is on one hand, it is going to stir by using a stirrer and it is going to provide oxygen, whatever is needed. So every knobs will be present, like for example, foam control, temperature control, pH control, everything will be present in the bioreactor itself. You just have to adjust and keep it. And whenever your product is ready, just withdraw it periodically. And whatever product is there, it's called as the culture. And you have to make sure you withdraw it what? Periodically, otherwise it will get damaged. Okay, so this is all about the process, how actually you produce your desired material from by using a recombinant DNA technology.